Good day to the viewers. Today we are going to discuss about classification of amino acid and its properties. Now let's see the introduction. Amino acids are essential to life and they are the building blocks of proteins. Some proteins function as enzymes while others serve as antibodies or support the structure. Proteins are composed of a number of amino acids linked by peptide bonds. The major key element of amino acid are carbon, hydrogen, nitrogen and oxygen. Each amino acid is a nitrogenous compound having both an acidic carboxyl group, a basic amino group and side chain representing R. R stands for the side chain that are different for each amino acid. R can be as simple as hydrogen atom or a methyl group or a more complex structure. The first carbon is the part of the carboxyl group and the second carbon atom to which the amino group is attached is called alpha carbon. The alpha carbon of most of the amino acid is joined by covalent bonds which has four different groups of atoms. About 500 amino acids occurring in nature, but only 20 amino acids form a part of the proteins in the human body. Properties of amino acids. Amino acids are colorless crystalline solid. It is generally melt at higher temperatures, often above 200 degrees Celsius. It is more soluble in water than in non-polar solvents. Optical activity. Amino acids having an asymmetric carbon atom exhibit optical activity. Asymmetry arises when four different groups are attached to the same carbon atom. All of the 20 amino acids except glycine are of the L configuration. Glycine is the simplest amino acid and has no asymmetric carbon atom and therefore shows no optical activity. All others are optically active. The mirror image forms produced with reference to the alpha carbon atom are called D and L isomers. The L amino acid occur in nature and therefore called natural amino acids. D amino acids are seen in small amount in microorganisms and as constituents of certain antibiotics like gramicidin, polymyxin, actinomycin and valinomycin, as well as bacterial cell wall peptidoglycans. Isoleucine and threonine have two optically active centers, therefore each has four diastereoisomers. Ampholyte. Amino acid can exist as ampholytes or zwitterions in solution depending on the pH of the medium. The pH at which the molecule carries no net charge is known as isoelectric point or isoelectric pH. In acidic solution, they are cationic in form and in an alkaline solution, they behave as anions. At isoelectric point, the amino acid will carry no net charge. All the groups are ionized, but the charges will cancel each other. Therefore, at isoelectric point, there is no mobility in an electric field. Solubility and buffering capacity will be minimum at isoelectric pH. Now move on to the classification. Amino acids are classified into four different ways based on structure, based on side chain, based on metabolism and based on nutritional requirement. Based on structure, amino acids are classified into different subgroups like aliphatic amino acids, aromatic amino acids, heterocyclic amino acids, and derived amino acids. In aliphatic amino acids, we have monoamino monocarboxylic acid which contain one amino group and one carboxylic group is present in the amino acid. Examples are glycine and alanine, and these are the simple amino acids. Valine, leucine, isoleucine are branched chain amino acids. Hydroxy amino acids which contain aliphatic hydroxyl groups in their side chain. Example serine and threonine. Cysteine, methionine are sulfur-containing amino acids which contain sulfhydryl groups in the side chain. Asparagine and glutamine are amino acids that contain an amide group. Amino acids with two carboxyl groups called monoamino dicarboxylic acids. Example aspartic acid and glutamic acid. Lysine, arginine are the example for dibasic monocarboxylic acid. Aromatic amino acids contain an aromatic ring in their side chain which contributes to their unique properties in proteins. Example phenylalanine and tyrosine. Phenylalanine contains phenyl group, a benzene ring attached to an alanine backbone. It is hydrophobic and nonpolar in nature. Tyrosine contains an hydroxyl group attached to the phenyl group, making it more polar than phenylalanine. It is polar and slightly hydrophilic. Heterocyclic amino acids are amino acids that contain a ring structure that includes atoms other than carbon, usually nitrogen within the ring. Example tryptophan and histidine. Tryptophan is an aromatic heterocyclic amino acid with a distinctive indole ring structure which is a fused double ring that includes a nitrogen atom. It is one of the essential amino acid which means the body cannot synthesize it and it must be obtained from the diet. Histidine contains an imidazole ring which includes nitrogen atoms. The imidazole group plays a significant role in enzyme active sites and helps in buffering as it can gain or lose a proton depending on the pH. An example for amino acid is proline. Proline has a unique structure where the side chain forms a ring with the amino group, making it a secondary amine. This causes proline to introduce kinks or bends in protein structure as it restricts the flexibility of the polypeptide chain. These amino acids are important because their structure influence protein folding, enzyme function, and the overall behavior of proteins in biological functions. Derived amino acids found in proteins. After the synthesis of proteins, some of the amino acids are modified. Example, hydroxyproline and hydroxylysine are important components of collagen. Gamma carboxylation of glutamic acid residues of protein is important for clotting process. In ribosomal proteins and in histones, amino acids are extensively methylated and acetylated. Derived amino acids not seen in proteins are otherwise called as non-protein amino acids. Some derived amino acids are seen free in cells. Example, ornithine, citrulline, homocysteine. These are produced during the metabolism of amino acid. Thyroxine may be considered as derived from tyrosine. Classification based on side chain. Amino acids having non-polar side chains. This includes alanine, valine, leucine, isoleucine, methionine, proline, phenylalanine, and tryptophan. These groups are hydrophobic and lipophilic in nature. Therefore, the parts of proteins made up of these amino acids will be hydrophobic in nature. Amino acids having uncharged or non-ionic polar side chains. Glycine, serine, threonine, cysteine, tyrosine, glutamine, and asparagine belong to this group. These amino acids are hydrophilic in nature. Tyrosine and cysteine may show hydrophobic character when present in the interior of the protein. Amino acids having charged or ionic polar side chains. In that we have acidic amino acids and basic amino acids. Acidic amino acids have a negative charge on the R group. Example, aspartic acid and glutamic acid. Tyrosine is mildly acidic. Basic amino acids have a positive charge on the R group. Example, lysine, arginine, and histidine. And based on the metabolism, amino acids are classified into purely ketogenic, ketogenic, and glucogenic, and purely glucogenic. Leucine is purely ketogenic because it is converted into ketone bodies. Lysine, isoleucine, phenylalanine, tyrosine, tryptophan are partially ketogenic and partially glucogenic. However, in humans, lysine is predominantly ketogenic. During metabolism, part of the carbon skeleton of the amino acid will enter into the ketogenic pathway and the other part enters into the glucogenic pathway. Then all remaining 14 amino acids such as alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, proline, serine, threonine, and methionine and valine are purely glucogenic as they enter only into the glucogenic pathway. And final one is based on the nutritional requirement. Amino acid is classified into essential amino acid, partially essential amino acid, and non-essential amino acid. Isoleucine, leucine, threonine, lysine, methionine, phenylalanine, tryptophan, and valine are essential amino acids. Their carbon skeleton cannot be synthesized by human beings, and so these amino acids are to be taken in food for normal growth. Histidine and arginine are semi-essential amino acids. Growing children require them in food, but they are not essential for the adult individual. The remaining 10 amino acids such as alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamine, glycine, proline, serine are non-essential amino acids because their carbon skeleton can be synthesized by the body. However, they are also required for normal protein synthesis. All body proteins do contain all the non-essential amino acids. In addition to that, selenocysteine and pyrrolysine are known as the 21st and 22nd amino acid respectively. They are less common than standard 20 amino acids, but they play a specialized role in some organisms and biological process. Selenocysteine is similar to cysteine, but instead of sulfur, it contains selenium in the form of selenol. Selenocysteine is found in selenoproteins which are important for antioxidant activity and redox regulation, and it is present in enzymes like glutathione peroxidase and thioredoxin reductase, both of which help in detoxifying reactive oxygen species in the body. Pyrrolysine has a side chain with a lysine residue linked to a pyrroline ring. This unique structure makes it distinct from the standard amino acids. Pyrrolysine is recognized as the 22nd amino acid and is used by some methanogenic archaea and bacteria in enzymes that are involved in methane production. Pyrrolysine found in certain methyl transferase enzymes which are involved in methane metabolism in methanogenic microbes. Thank you for listening the video.